let me start off by thanking everybody for showing up. That was very nice of people to go ahead and spend their evening with me. And what we're going to talk about today is gene and cell therapy and potential futures of gene and cell therapy. Can I ask you guys, if you happen to have cameras and you're not in some place where you're unwilling to be seen, it's really helpful to me if you turn on your cameras, and then I can sort of see my audience. Hello there. I got a wave from Sebastian. Thank you. I'm Jeff Galvin. I'm looking at my audience here. So to you, I look like a million years old. I am roughly, and I'm the CEO of American Gene Technologies, which is a company that is focused on this whole new emerging industry of our ability to go ahead and reprogram your DNA for better health. That's how I describe gene and cell therapy. And hopefully that's the way you guys think of it already. But if not, by the end of my talk, hopefully you will think of it that way. And some of you may decide, oh, I want to be gene programmers. And, what would a gene programmer be? It would be somebody that can design little constructs that can go into editing delivery mechanisms, viruses or CRISPR or whatever, that when they get into the body, they mitigate the drivers of disease, or they improve cells in a way where they can fight disease. And that is the way that I see this whole industry, because my background is that I come from Silicon Valley. I was in software, computers, internet, IT. When I was your age, I was getting my degree in economics and teaching computer science at the university as well. So I was really big on computers, and it's really interesting to think about why I was big on computers. Was I big on computers because I'm a computer nerd? Perhaps. I certainly became a computer nerd, but actually that wasn't what attracted me. What attracted me was that the first time I saw computers, I immediately saw this is going to be incredibly useful. This is going to improve lives. And how did I see it improving lives? I was like, all the most boring rote activities in life can be taken over by this thing that will tirelessly execute over and over and over again whatever software we put in it. And all we do is feed it electricity, and it will never complain. It'll just do it. It'll do it accurately, and it will never get bored. It'll never make mistakes so long as the software's right. And I was like, okay, even though it was back in the days where it was mini computers and teletypes, one of the things that I could tell was that I already seen mainframes. Now I saw it coming down to minis. There was the news of like programmable calculators coming out. I had a programmable calculator when I was sort of in my teens. So I realized this thing's getting smaller and it's getting better. And I was wandering around MIT on weekends, and I was seeing sensor technology and robotics. And I was like, there's nothing this won't do eventually. It'll drive our cars, it'll fly our planes, it'll build everything for us. It'll eventually be robots. It'll be small enough that we can hold it in the palm of our hand. It'll do, amazing, amazing things. Well, that's all come true. And I did spend 30 years in Silicon Valley doing that. And I was successful at it. So I ended up retiring at age 42. So 22 years in Silicon Valley doing that. And retired life is great. I recommend it for anybody. For me, it got boring pretty fast. So after getting married, which was interesting for a while too, and being retired on the beach, and that was interesting. So I had a place out in Hawaii and just relaxing. Then I wanted some more stimulation for my brain. When you're young, or let's say not old, and I would say that I'm not even old enough to be feeling like I don't want to do anything anymore. No, you want stimulation for your brain. You wanted to be doing interesting stuff. So what happened to me was I thought, okay, let's just sort of send out, the feelers for anybody that wants a board member, or maybe there's I can get back into tech in Silicon Valley by being a mentor for some entrepreneur, or maybe I could make a small investment in a company or whatever. And as soon as the word got out that I had money to invest, I started getting business plans from all over the country. And one of them came from the National Institutes of Health. right here in Bethesda, Maryland, from a lab that looked kind of intriguing. And the actual business plan was about DNA chips, which are, just these chips that can read DNA expression. So you can get a very quick look at somebody's DNA expression. And I thought, that looks useful. Getting a quick read on your DNA is going to be important in a lot of disease management in the future. So I flew out to take a look at it. Well, while I was out there, I got introduced to a guy named Roscoe Brady, who is incredibly famous, or was. He passed away in 2016, for curing Gaucher's disease. And a company called Genzyme in Boston was selling $1 billion of his enzyme replacement. Enzyme replacement is if you have a broken gene and you're not making an enzyme that you need in your bloodstream, which was the case with Gaucher's disease, it leads to some sort of manifestation. It can sometimes be a serious disease. And in the case of Gaucher's disease, it is an enzyme that allows you to metabolize lipids in your organs. And if you don't have that, your organs swell and they burst. So people are dead by the time they're your age because those lipids build up and they die. Anyway, he was able to replace that enzyme in these patients, but he thought it's got to be simpler than that. He thought, what if we could get them to make their own enzyme? And viral vectors was a new thing on the scene, and he was doing cutting edge research in lentiviral vectors. And he explained viral vectors to me, and I immediately just had this epiphany about the future of pharmaceuticals. So my epiphany was, and I said that to him right at the time, I was like, what you've just shown me is the ability to edit our DNA, that in a fully grown human being in the clinic, we can go in there and we can actually rewrite parts of the operating system of the human computer, the human cell, right? What is a cell but software, DNA, running in hardware, the cell, the cell mechanism, the cell machinery, and whatever's in that DNA gets executed in that cell. Now there's a manager in the nucleus that decides what part of the DNA to run, but how is this different than your computer besides just being more sophisticated? Your computer is all written in zeros and ones. It only works with zeros and ones. If it wasn't for A to D conversion and D to A conversion, it couldn't do anything. But we can actually take, through analog circuitry, we can take sensors and translate them into numbers that your computer can work with. And through digital to analog conversion, we can turn those zeros and ones into display, to display things on the screen, or servo motors that turn the wheel of your car, or control an aircraft or whatever, right? But that's what a digital computer is. It's all zeros and ones being processed by a machine that turns out other zeros and ones. Your cell is just amazing, right? It works in the materials it's made of. It's base 4, A, C, T, G. The 4 nucleotides that make up your DNA are like a base 4 instruction. And each one of the commands that's in there, each one of the genes makes a specific gene product. And then all these genes sort of interact with one another to make really sophisticated functions like, well, does your personal computer have an instruction that will actually make a copy of itself? Well, your cellular computers have that. An instruction where when you trigger it, the cell will split into 2 cells, right? That's how sophisticated it is. It doesn't just communicate in electricity like your computer does. It's electrical, chemical, cells can physically bump each other. You are nothing but trillions of collaborating organic computers that make up everything about you. That's how sophisticated it is. Now you're a computer where we didn't get the manual, right? So we have to figure out what all those genes do. But once we do, we're going to be able to do anything. And that was my epiphany as I was sitting there and learning that you can now crack open viruses, scoop out their viral DNA and throw it away. And then you can put whatever you want in there. And that empty shell that used to be the virus will carry in that good code to update your computer and make it stronger, better, and mitigate diseases, right? And I looked at this and I said, well, now that we can reprogram your DNA, what can't we do? This is the future of pharmaceuticals. The blind will see again. Cripples will get up and walk. The lepers will be healed. We could send radiation and chemotherapy the way of bloodletting and leeches. All right, now that was visionary back in 2007. And but that was what attracted me. It was just like the first time I saw computers and realized, oh my gosh, this is going to do great things for humankind. This is exciting. I've got to be part of it. So it turns out I wasn't really a computer nerd. I was a technology nerd. I love technology. I love things that lift us up, that make our lives easier and better. I love my cell phone. What can I say? It's just you guys don't remember the days before cell phones. But, we used to have to take phone calls at home, right? So, you could see that this technology, I could see the arc of it when I saw my first teletype. I could see it would be driving our cars. I knew we'd be carrying it around. I knew it'd be doing everything for us. Well, now what I can see is gene and cell therapy is going to completely change, completely disrupt pharmaceuticals. It's going to cure the incurable. It's going to treat even those things that were completely untreatable. It's going to make a future of medicine for you guys that your parents couldn't even imagine. They couldn't even dream of. It's going to, it's going to yield a cure for HIV. We're in a clinical trial right now where we have a what's called an autologous cell product. What that just means is we take some cells out of your body, we modify them ex vivo, put them back in your body. The cells themselves are the drug. What do? We pull out 400 milliliters of your blood. We go ahead and treat it in an automated cell processor for 11 days. After 11 days, we have 1 billion HIV-specific CD4 positive T cells that are immune to HIV. When we put those back in your body, you're immune to HIV for your whole life. Doesn't matter whether you have it or you don't have it. If we make that cell product out of your HIV T cells, and we can make it out of anybody that is HIV naive or anybody who has HIV that's been well controlled on antiretrovirals for 1 to 3 years so that they get their HIV T cells back. Once they have those, we can pull them out, modify them. We can expand them up to 1 billion cells, put them back in them. They will be not just normal, okay? An HIV-infected individual who right now has to take a daily pill to suppress that virus so that they don't get AIDS and they're not contagious. And that toxic pill means that they deal with diarrhea and headaches and fatigue every single day. But long-term, they get early aging, bone density issues, osteoporosis, brittle bones, liver, kidney, heart disease, and extra cancers. So even though you may think HIV is cured because Hollywood's not making movies anymore, and you're not seeing people that have the outward signs of HIV or the onset of AIDS, right? It's far from cured. It actually is a very painful life sentence for these people that are HIV positive, with a lot of diminishment to their just daily quality of life and their long-term quality of life and the cost of even just maintaining them. So, you think, oh, well, it's just $30,000 a year in pills. No, because they have $50,000 to $80,000 worth of doctor's visits to treat the side effects of those pills, right? All these things that I've been telling you about, all of these side effects. So this is a big deal. Something that was impossible 10 or 20 years ago is now really within reach. And we're in the middle of a phase 1 human trial for that drug process. And I can tell you right now, I'm 95% confident we have the cure. That at the end of this year, you'll pick up the newspaper and you're going to see, oh my gosh, HIV's been cured. And it's going to be this little known company called American Gene Technologies in Rockville. And you'll remember back to this day. But that's only the tip of the iceberg, right? Because gene and cell therapy could go ahead if you have a monogenic loss of function disorder. There's 7,000 of them out there. 1 in 10 people has a monogenic disease. Well, Gaucher's disease was an example. These people were taking painful injections of glucocerebrosibase every day to keep their organs from swelling up and from the organs from failing, right? But now we can just go into their bloodstream and we can replace that gene that they're missing, and their blood just starts making glucose ribosylbase. A one and done treatment. It's cheaper. It's a one and done cure, I should say. It's cheaper than treating them for life. HIV is cheaper to cure than treating them for life, and it's a much better life for them. All these monogenic diseases, all 7,000 of them, will eventually fall to the idea that gene and cell therapy has come to the point where I could get on the computer and order any human gene I want online, and it'll come the next day by FedEx in a plasmid, and I can be experimenting with this in the lab that afternoon. That's how far gene and cell therapy has come. It was driven by the fact that the US government made a massive investment in sequencing the human genome. Why was that important? Because we now knew what the genome was? No. It was important because that moonshot required so much equipment, so many PhDs, that what it did was it brought down the cost of everything, all the reagents, all the equipment. They came down because there was so much demand for it, because there was $6 billion worth of new work being done just sequencing. Now you guys send something out for sequencing, you know, you get it back immediately. You can sequence a whole genome for like $1,000. The first one costs $3 billion, right? That's the precipitous drop that we got. It was like what happened with the integrated circuit, the IC, the microcomputer. That was an event where the US government decided they were going to put calculators on every desk of the US government. And they went to Texas Instruments and they said, if we buy 5 million of these things, how much can you make them for? And they were like, okay, instead of $200, it'll be $2. And they did it, right? And that was what birthed a revolution in computing. Well, now we have a revolution in sequencing, in synthesis, in experimentation of knocking out genes, in viral vectors, in all these things that have come out of this whole burst of activity. We can cure monogenic disease. That's just almost embarrassingly obvious, right? You're missing a gene, I order it, I put it in a viral vector, right? So I take, what do we use? We use lentiviruses. What is that? That's HIV, cracked open, scoop out AIDS, scoop out the HIV genome, throw it away, clip some things in it so it's non-replicating. And now it's an empty stealth bomber that will deliver any gene to any cell in your body. Now we just need to decide what to put in there. Well, if you're missing a gene in your liver, we just go ahead and make some of this up with that gene. And that's our cure for PKU, phenylketonuria. Anybody here have a relative with phenylketonuria? Nobody? Wow. So lucky. 1 in 13,000 births, okay, in the United States. In the Mideast, it's 1 in 2,500 births. But what happens? They're missing one gene. They can't metabolize phenylalanine. Phenylalanine is in all animal proteins. It's in Diet Cokes. It's in diet sugars, all that stuff. The phenylalanine builds up in their bloodstream. It's toxic to their nerves and their organs, so they can never eat animal meat their whole life. They need special diets to try to replace the downstream products, because why do you have phenylalanine? Because phenylalanine hydroxylase breaks it down into a bunch of other things that you need. One of them is tyrosine, that's critical to brain health. So these kids are born without that. They have to go on a diet with no meat. They have to take supplements to try to slow down what? The long-term effects of being tyrosine starved in the brain. Tyrosine starvation leads to depression by college, right? Everybody has some friend that's depressed or has felt depression. Can you imagine how debilitating that is, to be guaranteed a depression, a constant depression by college age? Then it turns into schizophrenia by the time they're 30. They're hearing voices. By age 40, a third of them will have committed suicide because they're having suicidal thoughts from the schizophrenia. So it's like a slow death sentence, and it's just one broken gene in the liver. So what we did was we redesigned the gene so that we can get higher expression of it, because we need a lot of it in the liver, and it's expensive to make these viral vectors. We put it into a viral vector, we manufacture up the particles, we inject it into the liver, and the gene is replaced. Are they cured? Yeah, they're just not sick anymore, right? Because we have restored the normal pathways in their liver that process the phenylalanine. They aren't sick, they're normal now. We can do that for 7,000 monogenic diseases eventually. Then what are we doing with HIV? All we're doing is improving existing cells. Why do you get HIV? HIV is a very fragile virus. As a matter of fact, if you didn't have HIV T cells, it'd be exceedingly hard to get HIV. Why is that? Because it's the HIV CD4 positive T cell. The T cell that is responsible for initial detection of the pathogen. What happens is, in a cold, it runs over and it finds a cold viron. And the antigen receptor on that T cell gets activated. The T cell kills that viron. And in the excited state, it runs back to the lymph nodes and replicates. And it also, because it's a CD4 helper T cell, it's the conductor of the orchestra. It creates a CD8 response that captures all of the mutations and an antibody response that slows down the virus. And that's how you get on top of a cold. And a cold rips through your body in a way that a lentivirus can't. Lenti stands for slow. HIV is a lentivirus. This virus comes into your body, and if it's lucky, because only 1 in 10 times that you get a viron in your body for HIV, an HIV T cell comes near enough to it that that antigen receptor latches onto it and pulls them together. Well, what happens is that the viron for HIV has evolved the capability of grabbing onto the membrane and merging with it and taking its own DNA, the viral DNA, and installing it in that T cell. So the sentinel T cell that was supposed to detect the pathogen and start the whole immune cascade, instead has become infected and is now the beachhead for that HIV infection in your body. Runs to the lymph nodes and replicates, including replicating the disease. And it starts spewing out HIV virons, which then connect with all the other HIV CD4 T cells in your body and deplete them out. Within 90 days, you have 0 HIV CD4 positive T cells. Then your CD8 response goes away and your antibody response goes away. Now HIV has got control of your body. You're still not feeling sick yet. Still feel fine, because it could take up to 10 years before it depletes another T cell subset like a cold or a flu. And you detect it because you get a pneumonia that you can't get rid of. And they finally test your blood and realize you've got HIV. But that HIV CD4 T cell, it's almost like it was a Coast Guard helicopter that rescued the viron. Because if the viron is in your bloodstream for about 2 hours, it's going to be neutralized just by your normal serum deactivation that goes on in your bloodstream, right? But this CD4 T cell swoops in, rescues that thing, and then boom, you're infected. Well, here's what we do. We take your CD4 positive T cells and we just improve them a tiny little bit so that HIV viron can't get a grip. And if it can get a grip, it can't integrate. So how do we do that? Well, do you guys understand what siRNAs are? Short interrupting RNAs? Basically, we use those to shut off the message from a gene, right? So it turns out that one of the ways that HIV gets into your T cells is it grabs onto a receptor called CCR5, chemokine receptor number 5, and with another handle, it grabs onto CD4. Now, every T cell in your body has CD4, all the helper T cells, and they all have CCR5 as well, except for they found out that some people in Europe have a rare mutation where they're missing the CCR5. Turns out it's not a bad disease to have. It does have some minor side effects of extra skin infections, bacterial infections, and funguses, but you can get rid of those with creams and antibiotics and things like that. But without that handle for HIV to grab onto, the most common form of HIV cannot get a tight enough grip on the membrane in order to infect the cell. And those people are immune to the most common form of HIV. All right, well, what do? We slip into those HIV T cells an siRNA against CCR5. So we knock down the message, that cell loses that handle, we remove the door handle off that cell, it's harder to infect. We also put in siRNAs that are against specific genes that are actually in HIV, critical genes for producing HIV virons. It's called VIF and TAT. So we have 3 siRNAs that we're putting into each of these CD4 T cells. It's not just shutting off that door handle to make it harder to latch on, but if HIV gets into the cell, these siRNAs latch on to the genes and render them inert, unusable. And as a result, it further protects the cells. Well, that's all it takes. It's been proven that if you strip enough CCR5, that's enough to functionally cure somebody, make them immune for life to HIV without antiretroviral therapy. The dream come true for all HIV positive people. A one and done treatment where we modify their immune system so it's actually capable of clearing HIV. If your CD4 positive T cells work, you can clear HIV easier than clearing a cold because the HIV moves so slowly. And so a healthy immune system that's actually capable of fighting HIV will keep it suppressed and gradually eliminate it while leaving you functionally cured. You need no medication. You cannot infect another person. You don't have enough virus in your body to do it. I don't know if you've seen those ads, U equals U. Undetectable equals uninfectable. And it also can never progress to AIDS because the virus will never build up in your body enough to wipe out any other immune cells. And finally, you can never be reinfected because what's a little bit more HIV from the outside? It's nothing. Your immune system's capable of clearing the HIV that's in you. All we did was improve the cells a teeny little bit. That's another key aspect of gene and cell therapy. What's the final aspect of gene and cell therapy? I'm telling you a range from just replacing a gene that's broken over to right here, improving a cell so it can do its job, because that's all the problem is with HIV. HIV T cells can't do their job because the HIV viron has evolved one advantage. We take that advantage away, we improve those cells, you're cured. What's the last thing? We can modify cells so that they become little bots in your body that can do anything, right? The microbot, the dream of the microbot. And what is that? Well, CAR T. Have you guys heard of CAR T? CAR T is you take out T cells and you modify them so that they run after B cells and kill them. Now, why would that be useful? To have T cells that kill B cells. It's useful because B cell lymphomas are always in the B cells, right? So acute myeloid leukemia, acute lymphoid leukemia, right? Once the chemotherapies stop working, you're going to die from an acute case of leukemia. In the old days, they could just replace your bone marrow, right? But 6 out of 10 people die from a bone marrow transplant. So that's pretty extreme. Well, now what they do instead of replacing all your B cells with a new immune system, they go ahead and take some of your T cells out, modify them so they run after your B cells and kill them all. And what does that do when you kill all the B cells? Takes all the cancer also, because the cancer was in the B cells. Can you live without B cells? Yeah. You have to take antibodies every day. Is that better than dying? Yes. So what we've done, though, is this is the first attempt, and it's a successful attempt at creating bots in your body with a new function. Those T cells were not born to chase after B cells, but they found a little modification that made them a bot that was useful. And what's happening now is people that have acute myeloid leukemias and acute lymphoid leukemias are being cured without bone marrow transplants, right? That's huge. All right, now you go, wow, it's pretty high tech and it's going to be expensive, right? Is anybody thinking that? Like, will I be able to afford these cures? And the answer is yes. Can you afford a cell phone? Yeah. Could you afford an IBM mainframe computer? Your cell phone's about 10 times more powerful than the IBM mainframe. It's just a matter of time. Tech has finally come to biotech. They used to call it biotech, but it wasn't really tech. It was mostly bio, right? It was the same random discovery of things that you could put into your body that would be useful, right? Chemotherapeutics, biologics, small molecules, whatever. It's always random discovery, screen them down to something that's worth testing in a mouse. If it works, you might have a drug. $250 million later, you might have first human efficacy, and then you're rich. But only 1 in 5,000 molecules made it to that point. And why was that? Because it's all random, right? But now we don't do random stuff. We design stuff based on the knowledge of biology, the knowledge of genetics, and an ever-increasing ability to edit things with viral vectors, CRISPR, or whatever, right? We're at the nexus of this major planetary collision of system biology, our understanding of how does the immune system work? How do diseases move around your body? You know, just basically the mechanics of your body. And then genetics. How does genetics play in all of those things? What genes do what? How do they interact with each other? Remember, I told you you're basically a computer without a manual, right? That's what you are. We're discovering the command set. We're discovering what every gene does. We're discovering how those genes interact with one another in order to create you, your thoughts, your immune system, everything in you, maybe even your consciousness. We don't know, right? But the point is that this whole investment that we made in sequencing the genome, and now all the money that's pouring into gene and cell therapy and this whole industry of genetics is just made this incredible rocket, you know, trajectory of our understanding of genetics. And then in the meantime, quietly, all these ways of manipulating genetics have been developing. What are the ways of manipulating genetics? Remember, I told you viral vectors. We can put a gene inside a virus. CRISPR, we can actually identify a strand where it will actually knock out that strand and put in something else. Zinc-fingered nuclease, zinc-fingered talon, right? There's a lot of different ways to edit, and they all have different advantages and disadvantages. So what are those editing things? Are those like that? Is that the technology? No, technology is everything. Right? This is something we understand at AGT in a way that I think most of the industry doesn't understand. What's the difference between lentiviruses and AAV? Does anybody here know what an adeno-associated virus is? Or a lentivirus? It's just two different viruses, two different viral vectors. Well, there's a big debate out there. Should we be using lentiviruses? Is that the safest way to modify your DNA, or is AAV the safest way to? One of them's integrating, one is, you know, non-integrating, whatever. To me, this is like arguing between paper tape and punch cards, which one's best. Because all it is a software delivery mechanism. It's like the delivery layer of the old industry that I was in, right? One day we may be delivering cures over the internet. Why couldn't your cell phone at some point, you know, put it up against your body? It would go ahead and sequence your DNA, send it off to a computer. The computer would go ahead and calculate what's wrong with you, and it would create a replicating virus that you would inhale. It would sweep across your body and edit out your problem. That may be the way that the software for your human computer will be done in the future. That's a long way off, but we have these rudimentary ways of editing right now. And I was back in the paper tape and punch card days. Maybe rudimentary, it may be barbaric compared to what we're going to do in 40 years from now, but it's functional. We can create a ton of value out of it. The computer industry's been around for a long time. You don't have to be able to do everything in order to do something that's of value. What's the next layer up from that? The software? No. What we thought at AGT, what I thought when I met Roscoe Brady, is somebody needs to be the Microsoft of this industry. What do I mean by that? There's an efficient way to make a new drug, a new piece of software, and there's an inefficient way to do it. If I hand you an iPhone without an iOS, is that the way you want to write your next app? Writing on the hardware? No way. You need an operating system, a set of reusable tools and components that you can mix and match that can do 80% of the work, and then you can create on top of that. And that's the most efficient way to do what? Unlock the most value you can in gene and cell therapy, the quickest for the most patients in the most reliable way at the cheapest price. That's what we're doing. Would it blow your mind to hear that the cure for HIV only took us about $15 million to get it to this point? Remember, it costs about $250 million on average to get through a phase 1 study on a drug, right? In order to decide something that you want to test in a mouse, in these old random discovery of molecules and monoclonal antibodies and biologics and chemical entities and stuff like that, you normally had to screen 10,000 molecules for 2 years for a cost of $10 million just to find 1 or 2 of them worth testing in a mouse, and only 1 in 19 of those were worth trying to do a clinical trial. Think about the failure rate. Why is that? Because there it's random discovery. With us, it is design. We know what a gene does in your body. We know what shutting off that gene will do. We know what adding that gene will do, right? We even have an if-then statement. That's called a specific promoter. If I put a genetic construct into one of your cells, I can put a specific promoter on that construct that's like an if-then statement that can look at any enzyme or any protein in that cell and determine what type of cell it is. Is it the right type of cell that you're looking for? Like, I want it to only turn on in liver cells, or I only want it to turn on in B cells, right? So I have an if-then switch where that genetic construct will do nothing unless that if is satisfied. Does that sound like software to you? It is. And what else could it test for? A disease state. What if I only wanted to turn on in cells that have prostate cancer? I might look for human prostate-specific antigen. We have these specific promoters that will only respond to HPSA, human prostate-specific antigen. So this is, design. We can predict how it's going to work. The FDA will eventually be able to predict how it works. Eventually, we can simulate you in silico, and we can go ahead and test a lot of these genetic constructs in a computer before we test them in cell models at the bench, before we put them in mice, and before we put them in the clinic. So we've reduced already, without the in silico modeling of your body, which it's too early to do that right now. You're too complex, right? But we can still test everything in cell models in biosafety cabinets, right? In cell culture hoods. And that gives us a good indication of whether the gene's turning on correctly in the right situations. So we can debug our software at the bench instead of doing it in the clinic. So we have reduced the price of going from idea to first human efficacy from $250 million in about 5 years to 2 years and $10 million. Tell me a disease that isn't worth curing for that, right? If there's only 1,000 people that have a disease, but it's serious enough, you multiply 1,000 people times $50,000. What is that? 50 million? Is that correct? Or is it 500 million? 50 million. So see how dependent I am on computers? You see, computers are important. And I knew that as a 7th grader. So, but that's the point, is at $50 million, gee, that could be $20, $30 million in profit at $50,000 a dose. If you're going blind, would it be worth $50,000 to not go blind? There's a bunch of monogenic diseases that do that, right? There's one that's cured already, Leber's congenital amaurosis. A company called Spark put in a gene, RPC, I think it was. Turned out that these kids go blind in their early teens because they just run out of light-sensitive protein because their gene is missing that refreshes it. So they just put the gene back in. Blind people were seeing again. Remember what I said? Blind people will see. Spinal muscular atrophy. Kids born with spinal muscular atrophy don't develop a normal spine or the muscles. What does that mean? They die at age 4 in iron lungs, right? In machines that are breathing for them. Why? Because of one gene that they're missing. There's a cure for that now. It was made by a company called Avexis, and it was sold to Novartis. And a good percentage of those people have normal lives now. That's people that would have been crippled. They would never have been able to actually lift their own head, roll over, walk, any of that stuff. And now they're getting a life. It's the stuff of miracles. If we cure HIV, will that be a big deal? You bet it will be. I think we got a really good shot on goal here. I think you're going to read about it later this year. We're going to treat our first patient next month or May. Sometime in the next 2 months, we're going to treat our first patient. So we're going to be getting data on this. And since you're not allowed in gene and cell therapy, it's so cutting edge, the FDA won't let you treat it. They won't let you use it in anybody that's not sick. So we don't have to do the usual safety tests of what kind of dose can a college student handle that we're paying, a healthy college student that we're paying to take a drug to determine when it gets toxic, right? No, we're getting HIV infected individuals in this study, and we may see some of them get better, which means we won't just get safety data out of this, we'll get efficacy data too. That is a drug at that point. Anyway, that's the exciting future of gene and cell therapy. And will you be able to afford it? You bet you will. Where else can you see cell phones besides the United States? Africa. This industry is doubling in capability every year and halving in cost. What does that sound like? Sounds like Moore's Law. Sounds like computers. It sounds like, what the Intel guys thought about the power of computing every year. And we're seeing it, and I can tell you, because I've been in this for 13 years, we're going way faster than that. Doubling in half, we're more like 4Xing and 1/4ing. So if you look at the cost of sequencing, it followed Moore's Law for about 2 years, and then it fell off a cliff. Moore's Law would have gone like this, and this is at like nothing. And that is why, because computers are even useful in this. All right, so I think I should wrap it up there, because maybe you guys have some interesting questions. I mean, I've been blathering on. It looks like you're following me. Hopefully I haven't, bored anybody or lost anybody here. If you have some questions, I'm more than happy to answer them. And I guess the organizers of this have a little game that they want to play also.
Yeah, so first off, I want to thank you. This is super, super interesting. I can start the Q&A by asking, you mentioned how you can cure it for monogenic diseases, but how effective is this method for polygenic diseases?
Well, it's just a matter of being able to carry more genes, right? So how does paper tape compare to a thumb drive? Mm-hmm. Or a DVD? We're going to have editing capabilities that will carry, 50 genes. Why does it even have to be a virus or CRISPR or something like that? Who here has seen the movie Elysium? Anybody? So, nobody. Oh my gosh. Isn't Matt Damon cool anymore? I mean, what happened? All right. So I saw that movie, and one of the things that I thought was really intriguing is they have a machine in there. It's not really important to the plot, but you lie down in this machine. It has some like arm that just goes over you like this. And then it goes a second time, and then you're all better. And I was like, are we absolutely certain that your DNA cannot be read and written electromagnetically or with some sort of beam? I'm not certain of that. Could be. So I actually believe that one day that machine will exist. It is perfectly possible that this machine could just read your DNA, compute what's wrong, and fix it. And now they have it doing things like fixing broken legs. Well, why not, right? Because we could actually send things into your body that would go ahead and accelerate bone healing. We know how to do that. There are certain growth hormones that will heal your skin and your bones faster. So anything could be done with that. That machine could actually exist. So yes, would polygenic diseases be on the agenda? You bet. It's just a matter of when. This is what you need to remember. In an environment where the technology is doubling every year and halving in cost, what's possible? Anything. If you can dream it, you can do it. You may not know when you can get there, but you know you will get there, right? We will absolutely get there. And I think diseases during your lifetime, all the deadly cancers will be gone. I didn't even tell you about our cancer treatment. We have a cancer treatment where all you have to do is just put a little injection of viral vector into the primary tumor. It lights it up in a way where it excites your natural T cells that surveil for that type of cancer, wipes out that tumor, but since they're circulating T cells, it gets all of the secondary tumors and metastases at the same time. How would you like it if when you're middle-aged and you go down to the doctor and you find out you have prostate cancer or you have breast cancer, they're like, yeah, you're gonna need a shot, come back tomorrow. You're like, okay, but what about the surgery and the radiation, the chemotherapy? And they'll be like, what? What are you, in the dark ages, right? No, we just go ahead and we take a needle and we get it down into that lump that you felt. We go ahead and inject this viral vector in there. Your body takes care of the rest. The rest. No radiation, no chemotherapy, no 5 years of hormones. No, that is the future of deadly cancers. I think we actually have line of sight to that. It's a couple of years behind where we are on HIV, but I'll tell you, it looks just like HIV did to me a couple of years ago. I think that we've got something that's highly likely to make deadly epithelial cancers of every type of epithelial cancer, breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, and skin cancers. It has the capability of reducing those things down to a shot, making them a chronic disease. You get one, we fix it. You get another one 10 years later, we just fix it. You get another one 5 years later, we just fix it. And here's what's really interesting about the mouse experiments. We have 1,000 mice mouse experiment where 85% of the mice come back completely cancer-free from one shot into one tumor on a mouse that has multiple giant tumors of different types. One's breast, one's liver, one's prostate. You just treat one of them, you put it human gamma delta T cells in, and it clears all of them because it activates those gamma delta T cells in a way where they actually now have the eating capacity to eat away an entire solid tumor. All of us, everybody on this call has cancer. Every single one of us. No biggie. There's radiation, there's chemicals, all these things. You have T cells that are constantly surveilling, finding small malignancies and just eliminating them. Cancer never has a chance. But 1 in 3 of us will just get an unlucky event where we'll get escape. It's blowing up like a balloon. Now it's ahead of what these things can eat, right? Think about it, it's expanding geometrically, and these T cells just don't have the oomph. But we light that tumor up, and all of a sudden, boom, safely, these gamma delta T cells that clear those cancers, 3 to 5% of your circulating T cells right now, they've never been seen to eat the wrong thing. We can activate them up to the point where they will flush your entire body of epithelial malignancy. We treat your breast cancer. Turns out you had ovarian cancer, you just didn't know about it, mops that up too. We treat your prostate cancer. Turn out you happen to have had lung cancer as well, mops that up at the same time. Never even detected it. It's like a rewinding of your cancer clock. Very exciting. I mean, your future is just mind-blowing.
That's really interesting.
Talking on the cell phones and no longer being worried about disease. Sounds pretty good to me.
Mm-hmm. If anyone else has any questions, feel free to ask.
Oh, I'm actually interested. So, hey, Jeff.
Is that surprising? Somebody who's interested? Oh,
no,
okay.
it's like really interesting. Yeah, I think everyone was like leaving a message in the chat room that they're excited too. So I'm a instructor for genetic engineering class. So I feel like I have to pay you commercial fee for like telling students how excited like the stuff can become when it become reality, you know, like you talk about a lot of like interesting things in here. We introduced to our student about the construct concept that you talk about. We use a term biobricks and like let them know how you can mix and match like promoter with like gene of interest. So it's like really exciting. I feel like I have to pay you commercial fee for that. My question for you though, for the I know we don't have a lot of time, so I want to maybe jump right into the question. So
sure.
For the student in here, you know, like they're gonna either go to graduate school or maybe they want to get a job in the industry. So in my class, like I teach them like the basic concept of how things can be done with genetic engineering, with the recombinant DNA, how to use virus like plasmid, cell experiment, and so on and so forth, including the modeling that I think you kind of talk about that, like you can use experiment in cell culture instead of like human,
Yeah.
a lot of things like that. And then also bioinformatics with like MATLAB and all that. So what do you think a student who graduated from undergrad versus someone who has a PhD would do differently if, let's say if you have a job opening in your company or maybe company similar to yours, what do you feel like would be the most important skill they should pick up before they graduate so that they might get a job in a company like yours?
So if you want to go into the R&D lab, then you're going to have you know, sort of cell culture and pipette skills, right? I mean, that's the main thing, because that's where all the experimentation is done. That's where you test out your theories. That is your test bench to see whether the viral vector that you put together has the intended effect, and to also even do tests to see if there are side effects, right? This is your place to debug things, right? So let me give a pitch for everybody to take your class for sure. First of all you need is STEM background to get into what we're doing. You have to be a problem solver. You have to have logic. You have to know how to deal with facts. You need that in life anyway. But if you basically, I'm a computer programmer. I am the CEO of, I believe, the most advanced gene and cell therapy company on the planet. And why? Because I have a vision for what it can be, and I can espouse that vision. And that brought to me all the great PhDs, the masters and the undergrads, and everybody that could fill every position in this company, because there are so many things that need to be done in a company, and this can be done in a company. So your job could be anything from, you know, heavy duty R&D to translational R&D to regulatory, to marketing, to sales, to finance, to admin, to, I mean, we need everything here, right? So, you know, don't cubbyhole yourself. Decide what it is that you want to do every day, what you love to do. And trust me, it's being done at a gene and cell therapy company near you, because you need everything. It isn't just science. Science is 1/3 of what we do. Okay. You need all the pieces of the company. Everything has to go right for success. All right. Here's another thing why you must take, what is it, Dr. Chan's class? And here it is. Here is the reason. What if you had been just a casual computer programmer back in 1960, and you were like, I kind of enjoy this. I think I'll just get into computer programming. Okay, so first of all, you'd be like 100 years old now, right? But you'd still have job security. You'd still be in demand. Your technology would still be on this curve. If you kept up with it, man, you would still be programming amazing things. Why? Because there was a computer and software revolution. This revolution makes that revolution look small. Why is that? Everything attached to the internet right now, everything, computers, software, internet, networking equipment, your cell phone, smart devices, smart speakers, whatever, $3.5 trillion worth of sales per year. Healthcare, $10.5 trillion. The ability to create value in the body dwarfs our physical space. What would you rather, a fast car or a healthy body? What would you prefer, the next iPhone or to be alive to use that iPhone? Okay, the value in this space is mind-blowing. Anybody here heard of Goldman Sachs? Their prediction is this is a $5 trillion industry. Remember, pharmaceuticals today are only $1 trillion. They're like, it's going to 5x? I think they're underestimating it. There is no end to this revolution. First, we wipe out every disease on earth. Then we just start making you better. Better, faster, younger. How's that sound? You don't care because you're already young. But how would you like to be 60 and feeling like you feel today? Assuming you feel good. Also, remember, even if you're not feeling good, everything will be fixed up. So you'll be feeling good. But you can have your same skin, your same hair, your same muscles, your same lifestyle, right? Not a bad thing. That's the next phase of this thing. Then after that, it gets a little bit dicey. You're like, what do we want our baby to look like? Because that'll be possible, right? Go watch the movie Gattaca if you want to start thinking about that. That's a cool movie about that whole subject. But we get this image of what is human perfection and we start creating them. Before that, you'll be making new animals, right? Cows that don't feel pain. You know, maybe that'll be nice for vegetarians to know. Cow never had a thought, right? I mean, that might be, there's so many different things that we can do to improve life. This is an endless revolution. This is, in 60 years, oh my gosh, what we will do with this thing. But it'll be 10 or 15 trillion dollars a year worth of business, and it will touch every person on the planet because it'll get cheap enough and we'll pack enough value in it where they'll even be able to buy it. They'll be able to justify it even in third world nations because it'll be that valuable to them. All right, just like the cell phone. And you'll love your pharmaceutical company. You'll be working at a company that everybody loves. Why? Because although your drugs will be expensive at first, people will say, hey, that drug isn't, I'm not taking it because it's my only option and I'm going to die without it. No, they'll be taking it because it's an investment in their future, just like buying a computer. It'll be a consumer good. You'll love drug companies. You'll love AGT like you love Apple right now or Samsung. Why? Because we'll just be a competitive industry that keeps on obsoleting ourselves and coming up with better solutions at cheaper prices. And we won't be raping you all the time like the pharmaceutical industry does because you're desperate. Because if I raise my price of the HIV cure high enough, guess what will happen? Somebody else will come up with something that works that's cheaper. Why isn't the price of Excel $10,000? Because you go ahead and use Linux at that price or Google Docs, right? Everything is competitive in the software industry. This is a software revolution for the next 100 years. And you can learn all about it in Dr. Chan's class. So get over there fast before all the slots fill up. Because, you know, look, you can be whatever you want. I highly recommend STEM education. It'll make you a better citizen. You need to be able to determine a fact from a non-fact, and you need to be able to put in, you know, sort of logical processes in order to figure out what's true about the natural universe. And why? Because nature will eventually win. I like to say, I'm going deep into the philosophy right now, but reality takes place at the nexus of nature and human nature. Human nature has the ability to deny nature, to hold back nature, but eventually nature wins. And if you don't believe that, just look at 550,000 deaths from COVID. And there were a lot of people that were like, oh, you just need to dominate the virus, I'll be fine, right? You can't wish nature away. You need to know your world, and you need to be able to logic what happens when you act in that world. And you can go ahead and shape your future. And in gene and cell therapy, you could shape the future of millions of people by providing new functionality in DNA that improves their lives, that solves their problems, that gives them value that is only a fraction of what that value cost. And what does that mean? Everybody wins. You make money, the patient gets something that's worth more than what they bought, what they paid. Everybody wins. Society wins. If we cure HIV, who's the big winner? Society, right? Costs $100,000 a year per HIV patient, and we're getting 50,000 new ones every year in the United States right now. This is expensive stuff. And they don't, you know, they die in hospitals. They cost us money. All right.
Thank you. If you were convinced by that, you can take Dr. Chan's class, which is BME 304. I'm taking it right now. So and I know we're over time. So if anyone has to go, they can go. You can fill out the attendance form that's in the chat. Otherwise, I'm going to start the Kahoot. I'll share my screen. So if everyone's in, I can start. Okay.
That's a good turnout.
Okay, so yeah, so the first step of gene delivery is to specify the therapeutic DNA that targets the disease. Okay, so Jeffrey's winning.
Well, that must be a tie, though. I think if you guys do well on this test, I mean, I didn't exactly give you a lesson in all of these things. You could imply a lot of stuff out of what I said, but this is definitely going to give an advantage to people that are in this field.
So it looks like the students in BME4 are doing well. So.
Oh, do you get extra points by picking the answer fast?
Yeah.
Oh, cool.
I'm gonna read the questions. So what is cell apoptosis? I feel like we all know that. Okay, so genetic technologies install permanent modifications to which of the choices?
These are good questions.
Mm-hmm.
So that's interesting. I would have said the cytoplasm could also, sometimes vectors deliver to the cytoplasm, not to the nucleus.
Okay, so what is a lentivirus? Yeah, we talked about this before, so. So which one is not an example of a lentivirus?
Oh my God, only one person got it right. But...
That was a really hard one.
Yeah, it was.
Yeah.
But Anna's still on top.
Okay, so what is a monogenic disease?
Very good. That one, you just needed to know Latin.
Mm-hmm.
So how is immunotherapy not done?
Okay, this is the last question.
Oh, Anna. Oh my gosh, she was leading. Boy, things just changed. You have time to get it back.
Okay, so what do gamma delta T cells do?
This is a great question, but it's unfair because I left out this detail in my talk.
The majority got it. So. Okay.
Very good.
Okay, so Louisa, Sage, and Sarah, you guys are all winners. I think every one of you is in BME 304. So, and we will send the merch to you guys. We'll email you and stuff. But thank you guys for coming, and thank you, Jeff, so much for talking. It's really, really interesting.
And I just
It was
wanted to add
pleasure.
one thing. I'm going to send you an email with all of our information, our contact information. And if you want to follow Jeff or contact Jeff, if you could just, Aditi, help me forward that email to everybody so that I get a copy, that would be wonderful. Thank you guys so much.